Hello everyone. In this video, I am going to discuss one of my research work, that is development and characterization of Genistein nanostructured lipid carrier loaded topical nanogel for targeting melanoma cancer cell. So the contents will be introduction, objective, plan of study, materials and methods, and results and discussion. So, as we all know, melanoma is a type of skin cancer and it is the most dangerous type of skin cancer which affects nearly 75% of the population, and it occurs in several stages like stage 0, 1, 2, 3, 4, where 0 is the most preliminary stage and fourth one is most dangerous stage. And what like cancer, it is also having several treatment strategies like surgery, radiation, chemotherapy, immunotherapy, and targeted therapy. And for our research work, we have chosen chemotherapy. And for this, we have chosen one phytochemical, that is genistein, and it is an isoflavone phytocompound, and it has already been indicated for treatment of melanoma cells. So these are the physicochemical parameters of our drug, and from the log P value we can see that this drug is lipophilic in nature. So lipophilic drug delivery carriers can easily be prepared for its entrapment. So the objectives of our work are to develop and characterize nanostructured lipid carriers of genistein, then to develop and characterize genistein NLC loaded topical gel, and then we will evaluate these nanogel for in vitro anti-cancer activity and skin retention studies. So this is the plan of study. First, we have done pre-formulation studies, and for pre-formulation studies, we have done drug excipient compatibility, method development, selection of lipids and surfactants. Then we have prepared genistein loaded NLCs, and then we have characterized for various physiochemical parameters. Then genistein NLC loaded gel was prepared and it was evaluated for ex vivo studies. Then materials and methods. So the materials which we have used for our research work are genistein, solid lipid Pressirol, liquid lipid Labrafag, surfactant Poloxamer, and materials for gel is Methocel. So pre-formulation studies we have done using FTIR, XRD, and TSC. An analytical method we have developed using UV spectroscopy and our reverse phase HPLC. The formulation optimization was done on two variables, that are formulation variable and process variables, and these are the optimized parameters which we have used for our studies. So these are the various batches which were prepared, and this is the method. So solvent diffusion evaporation method followed by ultracentrifugation is used. So characterization was done for following parameters like particle size, zeta potential, polydispersity, morphology, entrapment efficiency, and in vitro release and stability studies. The in vitro anti-cancer activities was done by MTT So NLC loaded gel is prepared by this method, and it was further characterized for various studies, and ex vivo studies were performed on human cadaver skin. Results and discussion. So these are the results of drug excipient compatibility. Here we can see there are no compatibilities found in these excipients and drugs. Then calibration curve was prepared by RPHPLC, and it is found to show R square value of 0.99. And these are the batches which were prepared. Here we can see the G9 batch is having minimum particle size with maximum entrapment efficiency, and only this batch was used for further studies. The SEM microscopy images shows that particles were spherical in shape, and the same was proven with the help of AFM also. In vitro release studies show the prolonged release was shown with the NLCs as compared to the pristine drug. The stability studies also depicted the stable nature of these nanoparticles, and the anti-cancer studies revealed that the genistein loaded NLCs shows 15.37% enhancement in the IC50 value. The cellular uptake studies showed the retention of these nanoparticles in the skin cells, and the characterization of gel shows the enhanced occlusivity of this drug. The release studies from gel also were found to be prolonged. An in vitro occlusion test shows enhanced occlusion of this nanogel. And ex vivo studies have shown that the release of drug from nanogel is less, and, and the retention in epidermis is more. So these nanogel will be best for treatment. So in conclusion, we can see that in vitro anti-cancer activity was enhanced 15.37 times, and retention of genistein loaded gel in skin epidermal region is very high as compared to the normal drug dispersed gel. These are the references which we have used, and thank you.